I call this, what the heck is hydrolysis? So I want you to look at this particular sketch that I have made for you. And before we do anything, I want to dissect this word. Hydrolysis. Or maybe we could say hydro, and let's pronounce it correctly, everybody, lysis. Yes. If you ask me, nobody says it right. Because I'm lysing something, taking it apart with water. Hydrolysis or hydrolysis is how we make a big polymer into monomers. Hydrolysis is how we change a polysaccharide into monosaccharides. How we change a triglyceride into glycerol and fatty acids. How we change a polypeptide into individual amino acids. I'm just going to use carbohydrates, as my example here. So let's say I have a great big chain of glucoses. So this is 1, 2, 3, 4, 5, 6, 7 glucoses in a chain linked to each other. That's a polysaccharide. Some people would say an oligosaccharide, somewhere between 5 and 10, but it's definitely not monosaccharides. I cannot absorb these big molecules in digestion. I have to hydrolyze them. I have to cut them apart into monosaccharides. The way I do that is by adding water. So the definition that you should put in your own personal notes is that hydrolysis or hydrolysis is the breaking apart of chemical bonds by the addition of water. By adding water between these monosaccharides, I can make them come apart. I can make this long chain of glucoses into individual glucoses, a monosaccharide, another monosaccharide, like that. The way I do this is hydrolysis, breaking apart these chemical bonds through the addition of water. What I'm attempting to show you, I don't know how well here, in this section of my picture, is that I think of water as a scalpel that cuts apart these chemical bonds. Running the scalpel is an enzyme. So an enzyme that performs hydrolysis is called a hydrolase enzyme because it hydrolyzes something. Water is my chemical blade, and the chemical blade is driven by a specific enzyme that cuts apart my polymer into monomers, which I can absorb. I can't absorb polymers. I can absorb monomers. This is the basis of chemical digestion. is this process of hydrolysis where we snap apart these large food molecules using enzymes and water. Sucrase is a hydrolase enzyme that cuts apart a sucrose disaccharide into two monosaccharides. This is the idea that you want to have. And what I have done is given an example of each type of hydrolysis reaction that we use in chemical digestion. So we see here two monosaccharides linked to each other by what people would call a carbohydrate or a glycosidic bond here. That's this thing right here. The bond making two monosaccharides into one big disaccharide, two-part carbohydrate. An enzyme comes along adding a water into the middle of this thing. Only a specific enzyme that fits into this disaccharide, let's say it's the one I mentioned before, sucrase. Let's say the monosaccharide on the left is a glucose, the one on the right is a fructose. Here comes sucrase. The disaccharide is called sucrose. And the enzyme adds a water right here in the middle. One side gets the H from H2O, one gets the OH from H2O. And now I have two separate monosaccharides. I broke the chemical bond between these two subunits using a water. That's hydrolysis. For the biochemistry lovers in the audience, which I'm sure there are many, this is actually the opposite procedure of how I made the disaccharide. We made the disaccharide by taking water out of it, dehydration synthesis. We break it apart by adding water to it, hydrolysis. If we look at a lipid, generally speaking, a triglyceride. So here I have a glycerol or monoglycerol backbone, and then, fatty acids over here attached. There would be R groups here for the organic chemistry lovers, right? Depending on what fatty acid it is. And what we do is come into this thing and break them apart right here by adding waters. See how one side gets an H, the other gets an OH. And I get a glycerol and 1, 2, 3 free fatty acids. So glycerol, fatty acids, 3 of them that were making a triglyceride, and I used a lipase enzyme to hydrolyze this. The idea for proteins, similar. Imagine a couple amino acids linked together by what we call a peptide bond right there, and I add water to this peptide bond if I have the proper enzyme, and what happens? The two amino acids come apart. One side gets an H, one side gets an OH. Here's one amino acid, here's another amino acid, monomers that I can absorb. Is everybody with me? So a way to think about it, hydrolysis, as performed by hydrolase enzymes, is using water as my scalpel to cut apart molecules. The enzyme is the cutter, and The water is the cutting tool. Maybe a way to think of it. And this is how we perform these catabolic reactions where I chop large polymers into absorbable monomers. And enzymes are very specific as to where they will cut, what their substrate is. What do they cut? So carbohydrate aces, plural, will cut apart carbohydrates. Lipases will cut apart lipids. Proteases will cut apart proteins. And an enzyme and its substrate, as most of you know, have a chemically specific relationship with who will fit with what. So I'm speaking in big generalities here about what hydrolysis is. And yes, I actually do pronounce it that way in my mind most times. Generally, people say hydrolysis, but I'd suggest that you say hydrolysis so you remember you are lysing something by adding a water to it. And this huge bunch of digestive enzymes that you get to memorize are hydrolase enzymes. This is the way we do this. The enzyme puts the water into a molecule, splitting it into smaller molecules. So make sure that you look at your particular A&P textbook as to which hydrolase enzymes you need to memorize.